colleague Pietro Alano, who will discuss bioluminescent parasites as tools for drug and compound selection.
Sorry. Good afternoon. Well, exactly after what was discussed at the end of the last talk, the goal of our work was exactly to provide the community with a robust, sensitive assay, which would be scalable to really high throughput formats, because really I think we need to have in mind that basically the biology of gametocytes we know makes it quite difficult to predict what kind of classes of compounds we will ever find active against early and mature gametocytes. The way we started this work was really to bet on the very high sensitivity and the virtual absence of background of bioluminescent reporters to explore to exploit these properties, but also to explore a variety of other luciferase reporters which can populate the world of bioluminescent organisms. And the way we did, we started to produce transgenic lines with the gametocytes expressing a variety of different luciferases besides the Photinus spiralis, the firefly that has been used in HTS so far in falciparum. And we indeed found a couple of potent luciferases from the click beetle, which importantly were of natural variant able to emit in the green and in the red light. And to give you an example of how we could also exploit the versatility of these kind of reporters, we developed an assay which enables you to simultaneously read the green and the light and the red light from a mixture of parasites of different stages, which can be applied to any developmental stage of the parasites appropriately constructed. The bottom line is that you can basically mix early and late gametocytes expressing a green on the red luciferase and vice versa, treat them, and you see that the early gametocytes treated with chloroquine, which kills only them and not the late ones, have a drop in the signal of the green luciferase, and vice versa if the early gametocytes express the red one, while the mature gametocytes, the signal of the luciferase expressed by the late ones is virtually unaffected under chloroquine, and of course both disappear when you treat with epoxomycin, which kills all the stages. This is just to show you how you can productively deploy luciferase properties in drug screenings in malaria. But in the course of this work, we, in collaboration with the FIDOC, we produced a line in which we basically coupled our best luciferase with an old-stage gametocyte promoter, PFS16, integrated in a dispensable locus. And this line produced the luciferase, which we see as a MIG-tagged fusion in virtually, let's say, beyond 80% of the cells. And the potency of this luciferase enables us to give a good signal to background ratio, even using a few thousand gametocytes. And we started to use this line also to address the issue that has been mentioned before, that when you actually screen drugs against a non-proliferating cell, non-dividing cell, you are often, and the gametocyte is definitely the case, facing the case of the signal you are reading kind of persisting in dying or unhealthy cells. And that kind of has been a problem in several assays, because we are really not measuring proliferation, but basically viability to a certain extent. With this line, we explored this possibility of not using the commercial substrates, which always have additives and ATP to kind of give you a stronger signal, just by basically adding pure luciferin, the luciferase substrate, with no lysis solution and no ATP exogenous. And we see that although the signal is slightly lower, but is as stable as with the commercial substrates, the decay of activity of these luciferases in treated gametocytes is definitely faster. And this is quite important. If you bear in mind that, like, this is a gametocyte which is treated 24 hours with DHA, it's virtually indistinguishable from a healthy one, while its viability is down to lower than 40%. We want to visualize this directly, and you can do so if you actually put this culture on the bottom of a 34 well plate and put it in a black box, homemade, with a good CCD camera, and you basically just add luciferin on top of this layer, and basically gametocytes basically bioluminescent. This is a kind of single cell bioluminescence, which is kind of potentially very useful for several applications in co-culture, in other systems. This is just to convince you that these were not just dots, but they were actually gametocytes. But the important point is that if you treat these cells, like in this case with epoxomycin, you see that basically luminescence is away. You can do so with chloroquine as well and epoxomycin, and you see that basically the signal disappears if the gametocytes are dying. What is the point there? Is that really, with this formulation, you really are sure that the luciferase is expressed, and so the parasite has to make it, but also it is actually using the endogenous ATP. And we thought this was kind of an important improvement in the assay. Little summary of this point. Basically, I think we so far provided a line which has a kind of all-stage gametocyte-specific luciferase, quite a potent one. And David was mentioning our effort in a collaboration to actually now make a line which is expressing mature gametocyte-specific promoters. This is the work that Giulia Siciliano is doing in Rome, and we are actually at the point of having a line with a new promoter which is firing late in development to basically reproduce the same situation. This line makes stably good numbers of gametocytes, so it's kind of it's a handy tool. The potency of this luciferase enables to really reduce numbers if you really want to scale up things, and of course you don't have to do any purification step. This luciferin substrate formation also has the appreciable thing that is cheaper, and importantly results in a more reliable assay for screening compounds on a wide and large scale. At this point, really, the work has been done mainly in Donatella Taramelli's lab at the University of Milano. Sara, D'Alessandro, and Donatella got our line and started to use it in a few assays. This slide is quite complicated, is kind of heavy, but it is related to the work that has been done of screening the MMB validation set, which is a collection of antimalarials of a variety of chemical families. And this has been an exercise performed in the context of the Gates Foundation Gametocyte Consortium. And really the point I want to make here are like by columns and by arrows. By columns is the point that also David mentioned to you, that indeed early gametocytes are more sensitive to drugs, but still are not killed by several current antimalarials, while late gametocytes are definitely less so, and this is kind of obvious graphically by the shape of this histogram. The second point is in rows, and it says that basically the luciferase assay, based on the line I just showed you, and the gametocyte PLDH assay that has been developed in Donatella's lab, and it is published in this work last year, they really do have a very good correlation in terms of the result they achieve with very nice correlation coefficients. On overall, I also want to remark the high Z-prime factors, which obviously in HTS is a major point. This kind of correlation between luciferase and PLDH assay readouts is also explained in this kind of slide. This is the summary of what I found published in terms of the current malaria box, the 400 compounds, which has been mentioned before, screenings in a few labs on gametocytes. And in Donatella's lab, there was a further screening on late gametocytes with the PLDH assay, again with a fairly good Z-prime factor, especially if you compare it with the assays on mature gametocytes. And when these compounds have been, 37 out of 400 compounds showed up with a higher than 50% inhibition in this screening. IC50's values have been calculated, have been determined, and again, these have been determined in parallel by PLDH and luciferase assay with a very good correlation, which again means that those assays are fairly interchangeable. Of this kind of IC50 determination, 7 compounds proved to have an IC50 lower than 1 micromolar. These are the actual data, quite robust, I think, and confirmed by replicates. And again, when we compared how other assays performed, the ones that again have been published, and many more are not published, so I really look forward to have a confrontation of the variety of malaria box screening as a kind of evaluation of the different assays performance. But basically, it was quite nice to see that the 7 compounds that have been identified in this way are in many cases confirmed in terms of activity when the data was present by other assays, which is a good sign. I think this is not entirely comparable because the stages are different, so I kind of put them in a gray scale here. Interestingly, and I think we'll see if this will be confirmed, in one case, these assays have been able to detect also a male-specific compound, which is important. But again, I really look forward to having this chart expanding in rows and columns to really evaluate this assay performance. My last slide is really to kind of propose in a way that out of the evaluation of the several assays, I think we should propose a pipeline of gametocyte assays with the purpose of really going really high into HTS, but then with a good throughput, and then having kind of maybe a let's say lower throughput and more informative biology assays somewhere down in the pipeline. This should be, I think, discussed also in this symposium. I think this would be a perfect occasion. And from what we've been doing, I think luciferase reporters assays could be really a front line with an interchangeable with the PLDH, which has the potential important thing of being used on non-transgenic lines of a variety of isolates. Biologically rich, as they are called, assays can also refine the hits in this pipeline, preparing the luciferase SMA phase, which obviously needs to address the issue on mosquitoes. And if we have more wherever, I think it's very important to propose and discuss. My time is almost off, so basically this is the people who have been directly involved in the work in ISS, in Bologna for the luciferase, and in Donatella's lab, as I mentioned. And this is more generally our panel of collaborators, and this project has been funded by the Bill and Melinda Gates Foundation with this grant. Thank you very much.
Any questions? So, can you, following up on that pipeline, do you have any preference or do you have any suggestion where, I mean, how many essays do we need to do before we end up with the SMFA, which is the mother of all essays, apparently?
I think, well, difficult question. Maybe it's not a question for me, but
well,
I think we can.
You may have an opinion.
My opinion is that, I mean, frankly, I'm kind of pragmatical. If we have an SMFA which is a high throughput or the highest throughput possible, I frankly think that you can really have a really robust, sensitive frontline essay, which I would suggest to be luciferase, in my opinion, obviously, but I'm biased. And I think one could go to SMFAs, really, because I think we really want, I think it's, in other words, if we have a reliable essay as a front line, you kind of have a good confidence into predicting a kind of biological important fact, sensitivity. But of course, you know, this is my opinion.
Sure. Any further comments? If not, oh, yes.
Closer. So my question to you, Pietro, will be, what do you think about the use of transgenic parasite versus wild-type parasite? Do you think that at one stage this could alter the if not the readout, but the outcome? And do you see any bias or any good reason to be scared by having a different set of positives or hits obtained by one method compared to the other?
It's possible. I guess obviously depends where you place your reporters. The ILO1 locus from David Piddock's lab and Sanjita has been shown to be, as far as we can tell, dispensable throughout the whole cycle. But obviously, you know, you can never be sure. PLDH, you know, has the potential of being having a high throughput and could be the best kind of complementary assay for addressing this on, we saw now, new wild isolates or something like that. It's a very important point indeed. Thank you.
Thank you, Pietro.